Hello, my name is Delaney Medcalf, and I am a senior biology major here at USAO. And today I am going to be talking about my senior project over the retention of the oral microbiome across 3 familial generations. So just to give y'all a little outline about what my presentation is going to cover today. So I'm going to do an introduction into what the microbiome is, its roles within human health, the influential factors, and why I'm so interested in the microbiome, specifically the oral microbiome. Next, I'm going to go into my methodology, so how I did my sampling, what my survey methods were, what my survey results are, my future genetic analysis that I'm going to be completing, and then my discussion over my findings and my future expectations. So let's dive in into what the microbiome is, and specifically what the oral microbiome is and why it's so important. So the microbiome is a collection of organisms that live in and on the human body. And so the oral microbiome is specifically the microorganisms that include viruses, phages, bacteria, fungi that live in and on the soft and hard tissues within the mouth. And these specific microbial communities are, of course, a part of the larger ecosystem of the human microbiome, and they are impacted by a variety of factors such as diet, lifestyle choices such as nicotine, and marijuana, which have been found to cause oral microbiome dysbiosis, or the mix-up of microbial communities, and that can lead to periodontal disease and certain oral cancers, as well as family interactions. So, as I just mentioned, some of these microbial communities and bacteria have been linked to oral health disease and cancers and periodontal disease. And so that's why I'm interested in what the oral microbiome looks like within these family units and what could be potentially causing either this retention of the oral microbiome across familial generations, or why maybe they aren't the same across generations. And so there is a connection between familial microbiomes, especially passed between mother and infant, and that there's a direct connection between the oral microbiome between mother and infant. Literature has also shown that the affection rates between families impact the similarities between parental and infant oral microbiomes, and that affection between partners can also change the oral microbiome that, say, their kid might get. And so I have hypothesized that the oral microbiome within 3 familial generations will have high levels of retention and be more similar between family members that have higher levels of affection compared to those that have lower levels of affection growing up. So how did I go about doing this? So first, because I'm working with human samples, I had to go through the IRB training and approval process, and I had to present consent forms to each of my participants, and I used numerical codes and alphabetical codes for each generation to keep their identities anonymous when I am looking at these surveys and the future genetic results. The participants also completed a 25-question survey, which covered a variety of influential factors such as oral hygiene, diet, lifestyle choices, and this anonymous survey was completed through Canvas. I also made sampling kits to actually get the saliva samples, and that included 10 mils of mouthwash, which was placed into a conical tube and sealed with parafilm. Participants were asked to swish for up to a minute. There were some participants that weren't able to complete the full minute for a variety of factors. And then the mouthwash samples were placed back into the tubes and stored in the freezer until they were returned to me. And they were continued to be stored in the freezer until I could do DNA extraction at OU. DNA extraction was done at OU using 2 different kits. So my first 4 samples used the Power Soil kit, and the rest of my samples used the water kit, just because we did not have enough samples. We did not have enough of the supplies to use the soil kit for all of my samples. And then my last step will be to do genetic analysis. So my DNA extraction is complete, And so from here on out, I'll be working on qPCR, PCR, and then genomic sequencing using the Illumina MiSeq Nano Reagent Kit V2. So for my specific survey methods and questionnaires, so each person was asked to fill out the IRB consent form. So what that actually looked like is one of my participants was a student here on campus. So they came to me, they filled out their IRB consent form and asked me any questions that they had over what their research was going to look like. And then they went to the other generations within their family and they got the other two samples for me. So they were kind of the middlemen. And so what my generational alphabetical code looked like is A is for my eldest generation, B is for my middle generation, and C is for my youngest generation. And so each of these participants swished the mouthwash in their mouth for up to a minute, and then they were stored on ice or in the freezer until I could do DNA extraction. And then they also filled out the anonymous survey through Canvas that had 25 questions. And some of the specific questions are, please rate the amount of physical affection, including hugging, kissing, and close contact you grew up with. How often do you brush your teeth? And then how often do you visit the dentist? Do you use nicotine? Do you use marijuana products? If so, how often and what types of products do you use? Have you recently taken oral antibiotics? Do you have a pet? If so, does this pet sleep with you? And do you share food with them? And so some of these questions I specifically asked because there has been research that shows that if you have a pet and they sleep with you, or if you just have a pet in your home, you're more likely to have similar microbiomes to your pet. As well as with affection, there's a link between affection rates and similarities within microbiome, especially the oral microbiome, and then nicotine and marijuana have been shown to cause oral microbiome dysbiosis, which is the disruption of the healthy, normal oral microbiome, which can lead to an increase in periodontal disease, oral cancers, and things like that. And of course, antibiotics would completely, potentially wipe out the normal, healthy microbial communities in the mouth. So what I found from my survey was I had 32 participants complete this survey. I had 11 familial units who I took samples from. So I technically would have 32 participants who would have completed the survey, but one of my participants, the survey results didn't go through. So I'm in the process of getting that survey fixed. And so I only have 32 to talk to you about today. But out of my 32 participants, about 72% of them are on medications. And then I broke that further down into generations to see if there was a difference, and there was. And so about 91% of the eldest generation is on medication, and that averaged out to about 5 medications per person. Of course, that's an average, and so there were some people that took way more than 5 and some people that took way less than 5. I also looked at the middle generation, and 70% of the middle generation is on medication with an average of 4 medications per person. And then the youngest generation, which would be which would be C, has an average of about 1 medication per person. And one of the reasons why I think this generation is so low with medication use is because I had some of the youngest generation that were very young, and then I had some that were my age, and so there's a wide variety of age difference. And so if you have a little or a small child, of course they're probably not going to be on medications, whereas someone my age might be taking more medications. And so I think that's why there was a lower number of Generation C taking medications. I also wanted to look at antibiotic use, since that has the potential to completely disrupt the microbiome and any similarities that there might have been. And so I found that about 15.6% of participants had recently used antibiotics. One of the issues I found with this question was I didn't put a specific time limit on what recently looked like. And so I might actually have some people who maybe took oral antibiotics within like the last 3 months, but they didn't consider that recently. And so that is something that maybe next time around I will make sure that I do a little bit more thoroughly, is put a time limit on what recently actually looks like. I also wanted to look at the affection rate because that is one of the big things that I'm hoping to see if there's a correlation with my research. And so about 91% of family units, which is all three generations, have at least one affection rating of 5, which is all the time, or 4, which was often. However, I was really interested in seeing that only 2 out of my 11 family units had consistent affection rates, meaning that all three generations marked the same numerical affection rate for all three generations. So I only had 2 families that had all the same affection rates between all three generations. However, I did have 50% of my family units report at least a 5 rating, which was all the time, or a 4 rating, which was often affection rates, at least within one in all three generations. So that means that I had ratings of 5 or 4 with half of my families within all three generations. So that was interesting to see. So I'm interested to see what the genomic sequencing will turn out with these differences. So, like I mentioned, I only have my survey results. I still have some work to do at OU. And so, what this future genetic analysis at OU is going to look like is I'm going to be completing my qPCR, which will check for the presence of DNA, allow me to identify the proper primers for amplification, and allow me to know how many cycles I need to run for PCR to amplify the 16S ribosomal RNA. So, this 16S ribosomal RNA is important, and the RNA that I'm looking at because the 16S is a gene on the large subunit of the ribosome, and this specific gene is highly conserved within bacterial species, which means that all of the bacterial species have this 16S gene. However, this gene has a high susceptibility to variation. So even though all of these bacterial communities or species have this 16S gene, that doesn't mean that they all have the same genetic sequence, which allows me to look at which actual bacterial communities are found within each individual person and be able to look at the similarities across generations and maybe between family units. And so after I complete my qPCR, I'll be able to run my PCR, actually perform the genetic sequencing, and then finally be able to analyze my results and identify the microbial community structure within the individual participants as well as the family units, and be able to conduct an analysis comparing these genetic sequencing results to my survey results, and hopefully be able to see if there are any correlations with reported levels of affection across familial generations. So I have created and hypothesized some expected results. And so, as I've mentioned, I'm anticipating to see similarities within the oral microbiome between familial generations with higher affection rates, even if maybe those affection rates aren't consistent. And I am hoping that different individual communities will be present, and I can hopefully link that back to maybe lifestyle or diet choices, and also be able to see some retention of microbial communities across families. So what I did is I made this table that you can see where I broke down each family unit and made a hypothesis on if I expect to see retainment of microbial communities. Of course, this is a little bit difficult to do just because each individual person within this family unit maybe didn't use nicotine or didn't use cannabis or antibiotics, but because I am clumping them all together as a family unit, if one person used it, it counts as a presence, even if 2 of those generations didn't. And so, as you can see, I have family unit 1 is likely, family unit 5 is likely, family units 11 and 13 also is likely. And so what I did is I went through and looked at the affection consistency across generations, the presence or absence of antibiotic use, the presence or absence of nicotine, presence or absence of cannabis, pet intimacy, and if the diet was consistent among familial generations or not. And so why I thought these specific family units are likely is even though there's not affection consistency across these generations, I do think that because, they don't have the use of antibiotics in some of them, several of them don't have the use of nicotine or cannabis. that because, family unit 1 has no antibiotic use, no nicotine use, no cannabis use, no pet intimacy, that I'm thinking that there will still be some retention because even though there's not consistency across the generations, this family unit did have 4 or 5s on the affection rating scale. So I'm thinking that there will be some retainment and some similarities. With family unit 5, there was no antibiotic use, no nicotine use, no pet intimacy. And so I'm still thinking that there will be a potential for a likely of microbial community retainment, even though there was some cannabis use and there wasn't consistency across generations when it came to affection, just because some of these other factors are not present. And when it comes for family unit 11, I'm also thinking that this is likely similar for the reasons that I expressed for family unit 1. There's no antibiotic use, no nicotine use, no cannabis use, no pet intimacy, even though there may not be consistency across affection between these generations. There are not these other influential factors that have the potential to even have a greater ability to decrease this retainment. And the same goes for family unit 13, because they had very similar, if not the same, reportings as family unit 1 and 11. So when it comes to the groups that I marked as unlikely, that would be family unit 2, family unit 3, family unit 6, family unit 8, family unit 10, and family unit 14. So what I used to determine if I thought these were unlikely is I looked at the use of antibiotics, nicotine, cannabis, just because these influential factors I think might have a bigger impact on retainment than just affection levels. Because there are so many factors that go into the microbial communities and how they are present within the mouth. And so I think that some of those might be a bigger impact than maybe affection. And so with family unit 2, there was a presence of antibiotic use, and so I'm thinking that that antibiotic use may have the ability to potentially trump the consistent affection. And also, this family only had a moderate level of affection between generations, even though it was consistent. So I'm thinking that that might also play a factor in why they may not retain microbial communities. With 3, I think it's unlikely because one, there's not the consistency across generations. There's nicotine use, cannabis use, and pet intimacy. So I'm thinking that these factors will not allow maybe as many microbial communities to be retained across generations. When it comes to family unit 6, even though there is consistency of affection across generations, they have all of the other factors as present. And so I'm thinking that that will really impact the retainment of microbial communities across generations. When it comes to 8, I'm thinking that nicotine and cannabis use will impact this microbial community retainment. And there's also not that consistency among microbial communities across generations. When it comes to generation 10, even though there's not antibiotic use, there is no consistency of affection across generations. There is nicotine use, cannabis use, pet intimacy, and diet is not consistent. So I think all of those factors will combine together to allow me to possibly not see similarities between generations. And then when it comes to family unit 14, I'm also thinking this is unlikely just because there was antibiotic use and nicotine use. There's not a consistent diet, and there is not consistency when it comes to affection across generations. And so why I specifically put family unit 9 as very unlikely for retainment of microbial communities is because they have nothing really going for them, it seems like, when looking at their survey results. So they had no consistency of affection across generations. They had recent antibiotic use, they had nicotine, had cannabis, had pet intimacy, and did not have a consistent diet. And so all of these factors have the potential to mess with the oral microbiome and cause dysbiosis and potentially not allow similarities to be passed on through the generations. And so that's why I think that this group in particular will be very unlikely to have retainment. And so when it comes to my future work and what I would want to do after I get these results back with this specific study is I would love to expand my familial groups. So get more familial groups as well as potentially get some family units that have all three generations living within their household, because that is something that I did not have in this study. And so I think that would be really interesting to see if that is maybe a big difference with microbial retainment, is actually living in the same household. I also would like to do a more specific survey. As I mentioned earlier, after I kind of got my survey results back, I realized that there was some questions that I could have been more specific on, such as like, have you ever used nicotine? Because even if you're not using nicotine now, but say you were a smoker for 10 years before now, that's going to have an impact on your microbial communities and which ones are present. And so that's something that I would like to do, is maybe expand that survey, make it more specific. I would also like to potentially control for specific factors. So not have anyone in my survey who's using antibiotics, or only have people who have never smoked or never used cannabis, and also had people who eat a consistent diet, just to kind of control for all of these factors that are also impacting the oral microbiome, and maybe allowing me to have a more difficult time to tease apart if affection rates between generations is actually the causing factors, or if it is maybe one of these other influential factors. And so I would like to take a few minutes to acknowledge all of the great people that have allowed me to be able to do this research and even get this project going. And so I would like to thank the Lamar Lab at OU for allowing me to use their facility, use their sterile labs to do DNA extraction, to do qPCR, PCR, and allow me to do the genetic sequencing and analysis. And so Krithi and Chris have been huge helps. Chris has been in the lab with me every time that I've been there, making sure that I'm okay. And then Krithi has kind of helped Dr. Jones and I know what sampling methods to use, what to look for. And so I really wouldn't have been able to start this project and get it rolling had they not allowed me to use their facilities at OU. I, of course, would like to thank the people who volunteered their saliva samples to me, who did my surveys, and who let me use their samples for this research. And I'm really excited to see what I figure out after I get my genetic analysis done. And then, of course, last but not least is my mentor and advisor, Dr. Jones. She has been with me and helped me every step of the way, and helped me prep this presentation, get my paper ready, and just make sure that I knew what to do with my research and how to go about doing it. And so here are my references for my background and for my presentation. And then I would also like to just thank you for taking the time out of your day to watch my presentation, listen to the research that I'm doing. And of course, if you have any questions, please feel free to reach out to me at my USAO email. I'm more than happy to answer any questions that you have. And so thank you again for watching my presentation over the retainment of the oral microbiome across 3 familial generations. And I am very excited to see what my results come back and what my genetic analysis comes back with my results, and how affection impacts the oral microbiome. And so thank you, and I hope you have a great rest of your day.